Okay, so we just hit 2 o'clock. So Isabel, if you're ready.
Okay, good afternoon. Welcome to today's webcast, Implications of the COVID Pandemic on Cardiovascular Issues and Clinical Trials, a focus on biomarkers. My name is Jonathan Seltzer. I'm the Director of the Cardiac Safety Research Consortium, as well as serving as the Chief Scientific Officer of WCG. As we know, COVID-19 has had a profound impact on the clinical trial environment. For instance, there's a global challenge of monitoring safety remotely. Additionally, COVID-19's seeming interaction with the cardiovascular system, as well as serious effects on people at risk for cardiometabolic diseases, require additional efforts for cardiac safety monitoring in clinical trials. Because of this, the Cardiac Safety Research Consortium will be offering webinars which include the best thinking of regulators and scientific and industry experts about how to provide optimal cardiac safety for trial participants in these challenging times. Before we start the webinar, I'd like to thank our sponsor, WCG. Through WCG's IRBs, Copernicus and Verb, and over other 20 companies, they provide support and a variety of clinical development services to sites and clinical trial sponsors. So thank you very much for providing this webinar platform. Before we begin, for those of you who aren't familiar with the CSRC, or the Cardiac Safety Research Consortium, I'd like to provide you with a brief overview of what we do. The CSRC is an industry, academia, and government consortium founded through a memorandum of understanding with the FDA. It's multi-stakeholder collaborative partnership of industry, academic, and government leaders dedicated to advancing the scientific study of cardiac safety issues. Over the 13 years of our existence, we've provided leadership and guidance for regulators, public and private clinical researchers through an open, interactive forum to collectively address cardiovascular-related issues in clinical trials. Working together, we've published a great number of probably over 60 white papers with a public health objective in many of these different areas. We have ongoing, we're also sponsoring ongoing work in shock as well as pediatric sudden death, as well as whatever current issues come up. In that vein, the COVID epidemic has obviously impacted cardiovascular safety in clinical trials in a great way, and this is the sweet spot for the CSRC. Before we get started, I'd like you to meet today's speakers. We're fortunate to have really outstanding crew from in this topic. Preston Duman, who is the acting medical team leader, Division of Cardiology and Nephrology at the Center for Drug Evaluation and Research at FDA. Andy Farb, who's the Chief Medical Officer in the Office of Cardiovascular Devices at the Center for Devices and Radiologic Health at the FDA. From the non-regulatory representatives are two real experts in biomarkers. From Jim Januzzi is a Hutter Family Professor of Medicine at Harvard Medical School. And Kristen Newby, we're delighted to have Kristen join us. She's a professor of medicine and the co-director of the Cardiac Care Unit at Duke University Medical Center. The agenda today is going to focus on biomarkers. Preston is going to start us out by reviewing ongoing trials, repurposing drugs, evolving guidance, and evolving trial designs from the perspective of the FDA. And then Andy will give us the same sort of perspective from the Center for Devices. Jim will then talk about the effect on patients with cardiac disease and cardiac biomarkers, and Kristen will discuss the impact on clinical trials. There are a lot of slides. These will be available on the CSRC website shortly after this program. Also, if you have questions for the speakers, we will have a discussion section at the end, the last 10 minutes or so. There is a question box in the lower portion of the website you're looking at. Please submit any questions during that time, and we'll triage those to our speakers for the discussion section. So right now, I'd like to give a warm introduction to our first speaker of the day, Dr. Preston Dunman.
Well, thank you very much, and I hope everybody can hear me okay. My name is Preston Dunman, and I'd like to thank Dr. Seltzer for inviting me to be with you today. I'm going to ask forgiveness for my voice. I've got a bit of a laryngitis here today. I am an acting team leader in the Division of Cardiology and Nephrology at FDA's Center for Drug Evaluation and Research, which many of you know as CDER. I've been asked to summarize the various consequences of the COVID-19 pandemic on clinical trials, which from our point of view and our vantage point have been really far-reaching and profound. There's been an immense interest outside of the impact of ongoing clinical trials as far as new clinical trials to study COVID. And we've actually had an immense number of these come in and have a commitment to evaluating them quickly. But what we have learned in this early experience, we've actually applied to a final guidance document that's recently been published by FDA for investigators who want to study COVID, and I'll share that guidance with you here at the end. To begin, I'd like to create a picture for you of what we saw happening to all of our ongoing clinical trials in cardiology about 4 short months ago. What you see on the right of this slide is a typical phase 3 protocol that was ramping up sites globally in the 3rd and 4th quarter of 2019 for the investigation of a rapidly fatal cardiac condition. The bars arising from the x-axis represent the number of active enrolling sites, and the ascending line represents the number of subjects enrolled. What becomes very clear is that between February and March of this year, as the pandemic was sweeping the globe and various lockdowns and social distancing measures were being put into place to stem the tide of COVID-19 infections, enrollment in this trial, as with a multitude of others, essentially stopped. We in CDER understood the gravity of this slide as it represented a profound disruption of the clinical trial operations worldwide, and the likes of which we had previously, quite honestly, never seen. Beyond the effects of the pandemic on ongoing trial enrollment, the ramifications of what you are looking at in this figure cannot be understated. Patients were missing follow-ups, measures of efficacy were not occurring as scheduled to include images of various types, functional tests, and data acquisition from PRO tools. This issue of PRO data was profound, as switching the platforms by which this data is collected can impact the performance and the validity of the tools themselves. There were disruptions in the delivery of investigational medical therapies for the trials that were unique to the mode of administration. For example, delivery of investigational medical products that are taken orally might be accomplished by a delivery carrier. However, the delivery of therapies requiring subcutaneous injection or IV infusion would require home administration by qualified personnel, and the storage conditions for many of these products would require specific conditions to assure product stability. Let's see here, advance this, there we go. Yet our primary concern here was for subject safety. As trials disruptions resulted in patients missing various laboratories, vital sign and volume assessments, ECGs, and adverse event assessments at investigative sites. This was particularly problematic for investigational products with known on-target or off-target effects on platelets, coagulation competence, renal function, electrolyte balance, and ECG intervals, many of the drugs that we work with in our division, in fact, do. While FDA certainly did not have all the answers in early March when all this started, it was clear that we needed to fairly immediately engage the investigational community to recognize the gravity of what was happening and then to prioritize action steps to deal with these trial disruptions. So on May 16th, CDER communicated publicly that we well recognized the issues, that we understood the protocol modifications were going to be required, and that the deviations due to COVID-19 illness or COVID-19 control measures were going to occur. At this early juncture, we emphasized that the safety of trial participants remained paramount, and we recognized that each sponsor would have to evaluate each circumstance with the focus on maximizing participant safety while doing their level best to preserve trial integrity. We also communicated that documenting the reasons for these deviations was going to be important. In short order, sponsors began submitting written plans for trial stabilization programs to their INDs and incorporating measures such as off-site or in-home laboratory vital sign and ECG assessments, flexibility of imaging and functional testing schedules, and delivery of investigational medical product by mail or home health care services. Separate from the challenges of ongoing trials at the onset of the pandemic, there was the additional issue of encouraging investigations into new potential treatments for COVID-19, which is certainly in the public's interest to do so as efficiently and safely as possible. In response, a large number of PIND and IND applications to treat COVID-19 have been submitted to FDA, and there has been intense interest from the medical community in repurposing both investigational drugs as well as currently approved drugs to treat different manifestations of COVID-19 that may be contributing to its high mortality rate. The first wave of these proposals to come to our division generally involved attempts to tackle COVID-19 associated thrombosis, cytokine storm, and hypoxia. Working with other CDER division directors, our division director committed to a rapid turnaround sequence for all of these applications, targeting comprehensive, multidisciplinary reviews within 1 to 2 weeks. To achieve this end, Dr. Stockbridge streamlined the process for reviewing COVID-19 trial proposals in cardiology by committing myself and another senior medical team leader to do the primary clinical reviews of all of these applications. What became immediately apparent was that the absence of a scaffold on which to generate interpretable data, in the absence of that scaffold, a number of these trial proposals arrived as single-arm, uncontrolled, or historically controlled studies in mixed populations that would in fact generate uninterpretable results. To address this need for an overarching scaffold for generating meaningful data efficiently, CDER about 3 weeks ago issued a more comprehensive guidance titled COVID-19, Developing Drugs and Biological Products for Treatment or Prevention, and I've included the hyperlink to this guidance in the title of this slide. This guidance directly addresses some of the early issues we were seeing in trial proposals by specifically defining COVID-19 populations of interest. In this guidance, FDA recommends strongly that drugs to treat and prevent COVID-19 be evaluated in randomized, placebo-controlled, double-blind trials using a superiority design, and that the trials be monitored by an independent data safety monitoring board. The analysis plans of these protocols should include prospectively planned criteria to stop the trial for futility or harm. With respect to dealing with trial logistical and scheduling disruptions, the guidance makes clear that well-motivated changes based on information external to the trial can be acceptable, and that sponsors are encouraged to discuss these changes with FDA. Finally, the guidance makes clear that pharmacodynamic markers are well-suited endpoints for phase 2 trials. However, phase 3 primary efficacy endpoints, or endpoint composites, must demonstrate a meaningful clinical benefit in how a patient feels, functions, or survives. For COVID-19, this generally involves one or a combination of the aspects of the disease you see here to include all-cause mortality, respiratory failure, or components of respiratory failure, the need for invasive mechanical ventilation, the need for intensive care unit level care, the need for hospitalization, and objective measures of sustained improvement. So I'll finish up here with this slide that I wanted to provide you with the participants on some more important information links from FDA. This first link on this slide will take you to a site containing all 54 of CDER's current COVID-19 related guidance documents for industry, FDA staff, and other stakeholders. Please note that we have 2 additional guidances in progress. The first is a guidance for shifting PRO modes, mode of delivery mid-study, and the second is a statistical guidance for studies disrupted by the pandemic. I will make these additional guidances available to Dr. Seltzer to share with you as soon as they become available. I'll now turn this over to my counterpart for the Center for Devices and Radiologic Health, Dr. Andrew Farb.
Thank you, Jonathan, and thank you, Preston, for that introduction. And I appreciate the opportunity to participate in the webinar from another side of the FDA, the Center of Devices, specifically the Office of Cardiovascular Devices. I thought it would be useful to start with emergency use authorizations that many in the audience have heard about. And these are for unapproved medical products or new uses for approved products in the setting of the national health emergency. And these are targeted for conditions that are life-threatening and serious, for which there are no alternatives, that have a favorable benefit-risk profile, and importantly have evidence that indicates the device may be effective. This is a lower regulatory threshold than typical FDA approval. It's required that medical device reports and complaints should be submitted, and we can require some additional data collection. In the Office of Cardiovascular Devices, these EUAs have been primarily focused on remote monitoring tools and mechanical circulatory support devices used in ICU settings. And when the pandemic ends, these EUA devices will not be immediately pulled from shelves. There is a process under development for the rational withdrawal of some of these devices, and there will be opportunities to use some of the data collected on these devices to support new device approvals. So what are we seeing in the Center of Devices with regard to COVID-19? Well, on one end is the impact on clinical trial execution for medical devices under study. The highest priority that we share with our colleagues in the Center for Drugs has been on patient and research safety. Most ongoing device trials have been paused with some restarts in process. And the impacts have been profound, including delays in enrollment and trial completion, more missing or out-of-window assessments, transitions to remote health status monitoring and data collection, including mail, phone, or web-based platforms, each of which has their advantages and limitations. But we have taken the stance of getting the data is much better than missing data. Where we may differ somewhat from our colleagues in the Center for Drugs is that with devices, particularly novel devices or novel uses, there are considerable learning curve aspects to their use, and that is an element that can be very much impaired by trial pauses and delays because of the need to have the operators become experienced with these new tools. There are also risk-based approaches to clinical data and monitoring and audits in that it may be very challenging for monitoring staff to get to the individual sites. We have seen and we anticipate more updates to the case report forms, including assessments of COVID-19 status and exposures. The other side from trial execution is, of course, clinical trial analysis. And a lot will depend on where the trial is with respect to its time course. The impact's probably most pronounced on those trials that are midway along in either their enrollment or follow-up and now are facing a new environment of a COVID-19 pandemic. As we have seen, there have been some profound differences between what the event rates were expected to be when the trial was designed and what they are being observed at the present time. Certainly, patient behaviors that in response to the pandemic can change events, and these changes can be reflected in diet, medication compliance, and exercise. Changes in baseline covariates of enrolled patients may also be altered with more higher risk or emergency or emergent patients being enrolled because those of the patients are coming to their physicians while more stable patients may be staying away. We've seen sharp declines in physician visits, hospitalizations, heart failure admissions, and STEMIs, but on the other hand, increased out-of-hospital death rates, which may be COVID-related or may be due to other comorbidities unrelated to COVID. There will be CEC adjudication challenges with trying to determine causality related to the test device or to COVID or some combination. And then increasingly in device trials, we try to incorporate quality of life assessments, but these may be significantly affected that independent of cardiovascular events and may be secondary to social distancing and isolation. There'll be variability of COVID based on geography, the disease activity, timing, for example, what the patterns of disease are in New York City now versus they were 2 months ago, and of course the known impact of the pandemic on important subgroups, including minorities, the elderly, and those with comorbidities. And particularly important for device trials is the impact on differential impact on randomized controlled trials versus single-arm studies, where we don't have the benefit of a randomized control arm in which you might expect that the effects of the pandemic may be equally distributed. So that this will all lead to modifications of statistical analysis plans to account for COVID activity and exposure. You've heard from Preston about the guidance. I won't spend any more time on it, just to point out that there is a very useful FAQ section that gets into some of the practical changes that investigators, investigational sites, and sponsors can think about in modifying their trials as needed. So in terms of what we've been doing with regard to our interactions with trial sponsors, we've recommended that they maintain the original investigational plan and as much as possible and pre-specify any protocol changes to preserve study quality and validity. Some of the sponsors have been proactive and already instituted changes to their investigational plans, and for others, we are going for to reach out to them directly, voluntarily, to see if they need to change their trial designs and institute changes such to preserve the best quality trials as possible. This is a very dynamic environment, and it requires efficient and regular communications amongst the stakeholders that are involved in clinical research for regulatory review. And so the approach that we've taken is to be practical, logical, reasonable, and flexible. For cardiac biomarker assays and new applications in particular, you'll hear much more about this in the following speakers. Current labeling claims for the biomarkers are to aid in the diagnosis of MI for troponin and for the natriuretic peptides to aid in the diagnosis of heart failure. The current study designs are all-comer populations with presenting with suspected MI or heart failure, along with independent adjudication of endpoint events. and we can anticipate that COVID-19 will add complexity to subject enrollment and event adjudication in cardiac biomarker trials. And you'll hear much more about that from Jim and Kristen. So there is a potential silver lining post-COVID where we hope to see opportunities for new clinical trial approaches to make data collection more efficient and more patient-centered. We anticipate greater use of in-home evaluations, some of which will require validation, including telemedicine, use of PROs and questionnaires, digital tools, including vital signs applications and activity trackers, and in-home endpoint testing and imaging, for example, the use of the 6-minute walk test, or meter gait speed, biomarkers, and home echo. I think I recognize that we are still in the early stages of our appreciation and responses to COVID-19, and we are still on the clinical trials learning curve. So I will conclude right here, and thank you very much again for the opportunity to participate. I'd like to now turn the podium over to Dr. Januzzi to tell us about cardiac biomarkers and COVID-19.
Great. Thank you thanks so much. That was terrific, and I'm really grateful to be joining you all today. I want to thank the sponsor for having me aboard this wonderful group of speakers. My disclosures are here. So biomarkers are an interesting topic when we talk about clinical trials because they cover all facets of clinical trial execution, ranging from evaluation of novel biomarkers themselves, but also extending to how we perform trials, as I'll show you. So why are we even bothering to talk about this? Well, you know, the world is moving at the speed of electrons these days on the internet, and lots of information being shared with clinicians, researchers, regulators. And it's quite clear each day brings us something new about the SARS-CoV-2. In case you were watching the news today, we now may have a drug to add to our armamentarium for treatment. One of the earliest things that really sort of lit up the internet in February or so is the fact that COVID-19, the syndromic manifestation of SARS-CoV-2 infection, was marked by cardiac injury. So how and why might that be? Well, SARS-CoV-2, one of the ways that it invades, really the way it invades various cells in the body, is through binding to and hijacking the ACE2 receptor. ACE2 is found in a number of epithelial cells, including the lungs, the gut, as well as in the heart. So there are numerous theories as to the possibility that SARS-CoV-2 may be entering myocardium as part of its lethality. Speaking of the virulence of SARS-CoV-2 relative to the host, there's no question more than 50% of our patients that have died from COVID-19 are older, with a substantial percentage in nursing homes and other congregate settings. And there seems to be a predilection towards a higher risk for mortality among people with underlying cardiovascular disease. So if you just look at it in a univariable analysis, underlying coronary artery disease increase the risk dramatically. So whether that's cause and effect or shared comorbidities in an elderly population remains to be teased out, but cardiac complications seem to be rather common among patients with COVID-19. Among them, we see patients that develop myocardial infarction from underlying stress and or plaque rupture. There are case reports of viral myocarditis, definitely a high incidence of atrial arrhythmias in particular, as well as ventricular arrhythmias, possibly provoked not only by the infection, but some of the therapies that we give. And also, interestingly, perhaps a higher than expected frequency of stress cardiomyopathy, so-called Takotsubo syndrome, among patients with COVID-19. So we are indeed on the front line seeing a large number of various cardiac manifestations, and then of course the thrombotic complications that people have commented on and case series are now describing, including microvascular thrombosis, macrovascular thrombosis, and pulmonary thromboembolism. So a wide range of unusual manifestations from this rather complex syndrome. So naturally, given the fact that cardiac injury and stress are so common, it's quite well established that biomarkers are abnormal in patients with SARS-CoV-2 infection. But this is hardly a novel finding. For example, we can rewind over a decade ago where numerous publications, including those out of our own intensive care unit, showed that elevations in, for example, natriuretic peptides, as well as troponin, were actually quite common among patients with ARDS and influenza-like illnesses. So cardiac stress and necrosis is by no means something that SARS-CoV-2 owns. It can be seen in critical illness, and indeed, one of the very important take-home messages for those listening in today is that cardiac injury, regardless of mechanism, is quite common in critical illness. But why might that matter in the world of clinical trials? Well, natriuretic peptides and troponins are rather universally assessed in clinical trials these days, whether as an upfront inclusion criterion, as an endpoint, as a possible way to understand mechanism of efficacy of a therapy. And indeed, in clinical trials of biomarkers, obviously, if a marker causes elevation, if an infection causes elevation of a biomarker, that by definition may undermine the specificity of the application sought. So, for example, NT-proBNP in a heart failure study, if it's elevated in someone with SARS-CoV-2 infection, that's going to hurt the specificity for the gold standard diagnosis. I'm going to largely focus on clinical trials of drugs and devices. We utilize markers as inclusion criteria. Importantly, we use them for endpoint adjudication, so myocardial infarction as partially defined by an abnormal troponin, natriuretic peptides to define heart failure, for example, as well as toxicity and safety endpoints as well. A broad range of markers have been described as being abnormal. I have no problem with sharing any of these slides. We don't need to go through this exhaustive list. But it should come as no surprise to anyone that works in the biomarker space that, in the presence of critical illness with multi-organ system dysfunction, there's a cacophony of abnormal biomarkers that we and others have demonstrated in these patients. So the bigger question is how one might utilize these markers to evaluate patients. Well, there's a clearly established recognition, and the next few slides are just examples of this, of how elevated biomarkers might identify those patients that are at higher risk for need for ventilatory assistance, for mortality, for bad outcomes. So early data from Wuhan demonstrated numerous necrosis and stress markers associated with worse illness. Indeed, although the values at admission are typically not all that high, as one transits the hospitalization, those patients destined for complications on the left demonstrated a rise in natriuretic peptide concentrations, while those that had a more benign course may have had a bit of a rise during hospitalization, but by discharge, the concentrations had fallen. With respect to troponins, from left to right is increasing severity of illness. This is a figure in a paper that we'll have coming out in JACC very soon. And basically from left to right, we see the frequency of a myocardial injury as a function of severity of disease. On average, it's around 20 to 30% in all comers with COVID-19, but as is depicted here, as patients are sicker, the frequency of cardiac injury goes up substantially. And it's not just mortality, but also what we refer to as disease progression in the intensive care unit, which includes, among other things, ventricular and atrial arrhythmias, need for ventilation, etc. So it's a morbidity and mortality signal with these biomarkers. We have some unfortunate recent experience in the United States with this exact topic. This is a paper that's going to be published in JACC, or has been published online at least. As of the time that the paper was published in the city of New York, my hometown, there are more than 260,000 cases of COVID-19. That number as of today is over 405,000, which is more cases than every other country in the world with the exception of Brazil and Russia. So really dramatic run-up of cases in New York, and unfortunately the hospitals in New York City were subject to a large number of patients presenting with COVID-19 all at once. In this setting, especially in the era of electronic health records, we were able to gather troponin results on a large number of patients, just showing once again the predictable association between myocardial injury and adverse outcome. Much like with natriuretic peptides, a rising concentration of troponin was associated with worse outcomes than a static or low concentration. And lastly, again, as I mentioned earlier, among patients with cardiovascular disease, once again, those patients that are elderly with multimorbid presentations, the presence of cardiac injury in those with underlying cardiovascular disease was much, much more associated with adverse outcome than in patients without cardiovascular disease. So how do we find a path through this? Well, I mean, there are a number of different ways that we can look for solutions. One proposition has been, at least for clinical trials, to try to bring the testing of biomarkers to the home rather than having patients skip their study visits in the hospital setting, where institutions are largely putting up major barriers to patients coming in to be seen. There are important considerations from a pre-analytic perspective if you're going to do home testing. A number of different considerations that one needs to consider, need to think about in the pre-analytic space. In the analytic space, where are you going to run the samples? How are you going to run them? And what about near-patient options? Of course, there are near-patient options coming. This is a neat little device that's going to be used for point-of-care finger stick NT-proBNP testing. This, I think, will be a major solution for home-based testing for clinical trials of heart failure, for example, and is really on the near horizon. And then there's the post-analytic issues. If you're going to be testing in the home, how are those results going to line up with an automated assay that has maybe greater precision and a different reference range? And how are you going to get the results to your investigators and into the electronic data capture? So a lot of important considerations as we bring biomarker testing closer to home. And then one last point, as I'm about to get the hook anyway, is the fact that, to the extent that clinical trials across a wide range of different disease states biobank their samples, I am begging researchers across clinical trials, clinical trial enterprises to save their samples for a retrospective analysis of antibody prevalence among their patients, because we really, I think, will benefit greatly from learning about patterns of SARS-CoV-2 spread through the population, utilizing these biobanks that are collected for patients in clinical trials. So in summary, ladies and gentlemen, COVID-19, systemic infection, about 20% show myocardial injury or stress, but this is proportional to the degree of severity. So sicker patients have higher concentrations. Of course, a wide range of markers are abnormal in COVID-19, and many, if not most, are actually quite prognostic. And this is a really important point, and someone asked a question, and we'll get to this during the discussion. Elevated NT-proBNP and troponin do not necessarily, and in fact most often do not reflect heart failure or acute MI. And so as clinicians measure these markers, as trialists measure these markers, we need to think very carefully about how we interpret them. And lastly, the elevation in these markers may support clinical judgment in the context of logical interpretation. And in clinical trials, the same thing goes. So with that, I will stop and pass the baton over to my esteemed colleague, Dr. Newby. Kristen, thanks, and take it away.
So thanks, Jim. Happy to be here with you all. My task in the next few minutes is to go through a series of questions that we came up with that kind of tie in the things that Jim just talked about with clinical trials and how they may impact clinical trials. And I will show you the list of questions that we're going to talk through here. I think you'll find, and I think it's been highlighted well to this point, that there are probably more questions right now than there are answers, and I'll try to highlight some of the reasons for that, or at least re-emphasize some of the things that you've already heard. So let's start with the first question, and that is because of the disproportionate representation of patients with cardiovascular disease among the COVID-19 deaths, and this holds true for diabetes and high blood pressure as well. And these are data that are from the same study that Jim showed earlier. I've just pulled them out to kind of really highlight the point. Out of proportion to their representation among the COVID-19 cases, should we consider stratifying COVID-19 clinical trials by underlying cardiovascular risk? And just to quickly go through stratified randomization, it's a method of restricted randomization where we assign trial participants to a group or a stratum based on a baseline clinical factor or factors, hopefully as few as possible, and then we randomly assign to treatment within each stratum. And the purpose of doing that, really the main purpose is, although randomization should distribute baseline characteristics equally, there is the possibility that there could be imbalances between treatment groups for some of these known factors that influence prognosis or treatment responsiveness. And I think COVID-19 and cardiovascular disease is a great example of that. So the question is really, when should we consider stratification? When might this be useful for this primary purpose? And it mostly depends on the size of the trial. So stratification really is most important in terms of reducing type 1 error or improving power in small clinical trials. So under 400 patients, when the likelihood of these imbalances in baseline comorbidities is the greatest. But again, emphasizing that this is really important when there is a large effect of that baseline factor on prognosis. So we wouldn't do it if there was a minimal effect of a factor on prognosis. It also could be important when we're doing larger clinical trials where we have interim analyses that may come earlier when sample sizes are smaller, and again, have the same effect on minimizing these imbalances. Other reasons are to facilitate subgroup analysis at the end of the trial, although that's not a critical need. And then in equivalence trials, it may actually help to reduce sample size, although it's rarely used for that purpose. So the second question was, how should potential clotting or myocarditis, and I chose to focus on clotting in the interest of time, but I think it highlights the challenges that you've just heard discussed when we're thinking about individuals with COVID-19 and doing trials in COVID-19. And that is that the two things. One is in clotting, for example, the range of manifestations and drivers of thrombosis, whether it's arterial or venous thrombosis, are quite broad. We have a population that has the traditional risk factors that we think about for venous thromboembolism. For example, they are bedridden, immobilized. They have heightened inflammation that may cause changes at the endothelial level that predispose to arterial or venous thrombosis, which is manifest by elevated cytokine levels and other markers of inflammation like IL-6. There are a number of hemostatic abnormalities that we see that range from microvascular microemboli in the pulmonary or cardiac circulation to direct injury. And you heard about the elevation in the cardiac biomarkers, the natriuretic peptides and troponins. But we also see pretty commonly elevations in things like D-dimer and fibrin degradation products, the prothrombin time, and often thrombocytopenia. And this may be reflective of a more systemic, disseminated intravascular coagulation type picture. And then the clinical manifestations that are surprisingly frequent in COVID-19 are things like DVT and PE, acute coronary syndromes, or evidence of myocardial necrosis or injury, as Jim highlighted. And then I mentioned DIC. I think one of the challenges that we have is shown on the right-hand side of this slide, that where we have the most information is in hospitalized patients, and those patients tend to be sicker. They have more of these risk factors. They tend to be the ones that we're measuring these biomarkers in more frequently. So our data are heavily weighted towards these more severe illnesses. And I think there's a lot to learn potentially from the asymptomatic populations and the more mild populations in terms of the prognostic relevance and how we use these biomarkers as we're thinking about evaluating patients and potentially using it for guidance of management. So if we think about evaluating for clotting, I think we can think on the venous side and we can think on the arterial side. For hospitalized patients, it's pretty standard practice that we have a CBC, so we'll have a platelet count, we'll have coagulation parameters, and likely in COVID-19 now a D-dimer at admission. So we do establish a baseline when we know these markers are associated with severity of illness, and they tend to be more profound as the illness progresses. I think repeat testing in the setting of change in status is probably helpful. There are also risk stratification models if we're thinking about venous parameters that we can also use, and these may help us understand the need for therapeutic anticoagulation versus routine venous thromboembolic prophylaxis. If a DVT or PE is suspected, there has to be a high index of clinical suspicion, again, because of all the baseline biomarker abnormalities in the population that we're most likely to see a DVT or PE in. So things like typical symptoms, disproportionate hypoxemia relative to the chest x-ray or otherwise the clinical status of the patient. RV dysfunction may be helpful. D-dimer, again, because it's often elevated already, may not be as helpful, although if there is a baseline for comparison, an acute elevation or very high levels may change our index of clinical suspicion. And then outside of the setting of COVID-19, cardiac troponins and natriuretic peptides are useful both prognostically and in thinking about treatment, but may be less relevant in this population. I think we have to really think about their utility. The echocardiogram, I think, is probably one of the strongest tools that we have to assess for RV dysfunction, particularly if PE is suspected. And we have to remember that in these critically ill patients where PE is most likely, many of our diagnostic tests, PE protocol, CTs, and certainly VQ scans, likely will be impossible. And so oftentimes whether it's for diagnostic purposes or to establish an endpoint or just for clinical care, we're making presumptive diagnoses. So these are, I think, challenges that apply not only to clotting, but to heart failure, myocarditis, myocardial infarction, et cetera, as we think about clinical trials in the era of COVID-19. If we're thinking about suspected ACS, whether it's in a COVID patient or not, the diagnosis itself for the initial evaluation includes the 12-lead electrocardiogram, and echocardiogram may be very helpful to look for wall motion abnormalities and potentially spare PPE use if the clinical syndrome is not clear. But for ST segment elevation, hemodynamic instability, and or focal wall motion abnormalities, most would proceed to acute angiography. Just to highlight, serial troponins may be helpful to look for acute changes, but remember that they're, again, often elevated in a quarter to a third of patients, again, depending on where they are in their illness severity. So it may be less helpful. And we're also finding things like Takotsubo's or microvascular injury presumed based on the absence of obstructive coronary disease, even when ACS symptoms seem typical. For DIC, there's really not much difference in DIC. It seems to be fairly common, but it's probably not unique to COVID-19. There are calculator scores, and we monitor the usual things: platelets, D-dimer, and fibrinogen. Should we get cardiac biomarkers in all COVID-19 trials? I think the answer is yes. I think we need to think carefully about how we're going to use them, but I don't think if we don't do it systematically, we won't learn about this disease, and we won't be able to put them in context and understand them across the spectrum of severity of COVID-19. So certainly they are useful for establishing a baseline, but the interpretation and their utility really depends on why we're testing them. Very prognostically important, maybe challenging from a diagnostic or endpoint definition standpoint, but they may help us understand treatment effects. And if we do measure them, we need to do it systematically. Potentially, they could help us understand cardiotoxicity, but it depends really on the phase of the illness in which we're testing a new therapy. What about other biomarkers? The same issues really are at play here. Abnormalities in inflammation, necrosis, coagulation, hemodynamic stress are common, and so it's really about why are we testing them? Can we establish a baseline? How do we interpret them in the context of clinical status? And can we use serial testing to inform our understanding of disease course, what acute changes mean, and or treatment response? But if we do that, we have to do this systematically across all phases of the spectrum of illness, and again, with interpretation in clinical context. And then finally, Jim raised this at the end of his talk, but this question about whether we should collect samples and store them for later SARS-CoV-2 serology. I personally think, and I suspect Jim would agree with this, that it's reasonable to collect and bank biospecimens in general in the context of clinical trials. They may help us understand the disease course, treatment effect. I would highlight that at least in the setting of COVID-19, there's no clear role for using biomarkers specifically to guide treatment, but I think that may evolve over time. The role for serology in specific, whether it's clinically or in research, I think is as yet not entirely clarified. We can think about banking samples from trials other than trials of treatment of COVID-19, so maybe in our cardiovascular clinical trials. And I think the importance here is that we are now learning that somewhere between 40 and 60% of infections may be asymptomatic. We don't understand what the implications of a prior infection are on a specific disease, its outcomes, resilience to subsequent cardiac events or other events, or on disease course. And it also may provide some estimate of the prevalence of prior infections that may be important in understanding the outcomes of a trial. I will highlight some of the limitations of the current generation of assays. They're mostly qualitative at this point, but there are evolutions on the way and becoming available that I think will make this more relevant. We do also need to be cautious of the specificity and false positives in the setting of what is a relatively low prevalence of COVID-19 at this point, although that will change as population prevalence of infection increases. In acute COVID treatment trials, where we're treating people who are infected, we know antibodies develop about 2 to 3 weeks after infection. They may occur as early as a week, and I think here we need to think about the type of antibodies, whether they're specifically looking for neutralizing antibodies. The extent of the antibody response may be important, and the duration of the antibody response, I think we still don't understand. So there may be opportunities there by collecting and banking samples, both acutely and during follow-up. They may also have some relevance for late complications. For example, the multisystem inflammatory syndrome in children may be a late immune response. And then obviously in vaccine trials, it's critical to understand the generation of the immune response, the persistence of the immune response. And it may also help in selecting people for vaccination, where you really want to vaccinate people who don't have evidence of prior infection. So I'm going to conclude there, and I think turn it back to Jonathan for discussion.
Yeah, thanks very much. I want to thank the panel so much. It was a very, very rich discussion. A lot to think about. We had, I'm looking at the numbers of people who stayed on, and it's a very, very sticky audience, which is compliments to all of you. We have only a couple minutes left for questions, so I'm going to take the liberty of just picking one and ask each of you sort of a 30-second response if possible. And we've sort of been dancing around this topic the whole time. I'd just like to maybe Jim, or actually I'll go in order, the reverse order. Start with Kristen, Jim, Andy, and Preston about how should we define COVID-19 associated cardiovascular events? Just if you could just take a few seconds and give your thoughts about how you'd like to see that as a regulator and as an investigator or a scientist.
Sure. So I'll start. I mean, I think again, the challenge is just the non-specificity, and I think we're going to have to be incredibly clear what event it is that we're looking for. So, for example, if it's myocardial infarction, and it's complicating COVID-19, I think it's probably going to be a rare event. And I think, we have to recognize that the majority of those elevations are not going to be related, and we're going to have to rely on our typical symptoms, other clinical signs, and, potentially additional testing. If we want to understand simply myocardial injury, I think we have a couple, I think that's a little bit easier. We look for changes from baseline in the typical cardiac markers, but that doesn't really help us with the etiology of those elevations, and so I think that becomes the challenge.
Okay. Jim, Preston, Andy, anything to add to Kristen's thinking?
I completely agree. An MI is an MI. It's defined by strict criteria, including the presence of coronary ischemia. Heart failure is a syndrome, but it has ways to define it. The challenge here is making sure that clinicians at the bedside do not conflate abnormal biomarker results with the presence of an otherwise clearly defined syndrome. We not infrequently see, elevated troponin, probably type 2 myocardial infarction. Wrong. A type 2 MI is a very specific definition. And so it's inherent on us as trialists as well as clinicians to get the definition accurately, which is why, the ESC, ACC, and AHA have all come out and said clinicians should be very cautious about ordering and interpreting these markers at the bedside in order not to mistake the correct diagnosis, which can actually mislead trialists when they're trying to interpret source documents from sites that have patients that were hospitalized with COVID-19.
So Andy and Preston, does that mean that can this be protocol driven, or does that mean we're going to have to adjudicate all these potential events, or some combination?
Yeah, this is Preston. I couldn't agree with Kristen more as far as the use of these and using them as part of a defined clinical syndrome, particularly the acute MI, where there are criteria. I think in heart failure, their use there is going to be, I think, to be determined by data from clinical trials where they're actually looked at sequentially and then correlated with carefully and in a systematic way with the outcome of these patients in the intensive care unit.
Andy, any closing words, closing thoughts?
Yeah, I think this may be the time that CEC charters need to be looked at and updated. We're still relatively early, and we will be learning a lot. And then the idea that to try to assure some consistency among trials to get the stakeholders and experts together to try to come up with reasonable and helpful definitions for trials going forward. But right now we have to deal with the data that we're generating in hand and putting the burden on our adjudicators in the CECs.
Great. Well, I think that's a great ending, and it's probably something CSRC should be able to take on, is to help create some of these definitions as our knowledge base grows. I want to again thank everybody for their participation. Unfortunately, we don't have time to continue the conversation. CSRC will be having another webinar in our series on implications of COVID-19 pandemic, talking about something that Preston and Andy talked about, remote monitoring of cardiac safety, also Jim, practical and regulatory issues. This will be early in August of 2020. If there's any follow-up questions, I'm sorry we couldn't get to them. You certainly can contact us at info@cardiacsafety.org, and we'll try and get back to you. On that, I'll thank the presenters once more. Thank you to WCG for hosting us, and everybody stay safe. Thank you very much.